Let's talk about apoptosis. The process of programmed cell death that occurs is a multi-step pathway in every cell in multicellular organisms. This is a normal part of any cell's life cycle. Carl Vogt, a German scientist, was the first to describe this principle behind apoptosis in 1842. In 1885, Walter Fleming, an anatomist, honed down on a more precise description of the process of programmed cell death. John Foxton Ross Kerr was able to find the difference between apoptosis and necrosis.
Apoptosis is different than necrosis because necrosis is a form of traumatic cell death that results from acute cellular injury. Apoptosis is the process of programmed cell death that occurs as a multi-step pathway in every cell in a multicellular organism.
Although Kerr was so important, we do not have a photograph of him because he was afraid that the camera would steal his soul. Kerr published his findings and was soon joined by Alistair R. Curie and Andrew Wiley. In 1972, they published an article in the British Journal of Cancer. On March 14, 2000, Kerr and Robert Horvitz received the Paul Ehrlich and Ludwig Darmstadt Prize for their description. Sidney Brenner, Robert Horvitz, and John E. Solston won the Nobel Prize in Medicine for their work in 2002.
Apoptosis occurs every day. Up to 70 billion cells die each day in just the average adult.
This represents a normal cell. Let's go through the different cell parts. Cell wall, receptors, mitochondria, ribosomes, Golgi, nucleus, rough ER, smooth ER, centrioles. Alright, cool.
In apoptosis, the biochemical events that lead to the many physical and chemical characteristic cell changes and death occur through the cell's morphology. The changes that occur are bleeding, cell shrinkage, nuclear fragmentation, chromatic condensation, and chromosomal DNA fragmentation. This process is controlled by a range of diverse cell signals that originate outside or inside the cell. Extrinsic inducers, such as toxins, hormones, growth factors, nitric oxides, and cytokines, must cross the plasma membrane or transduce in order to have a response. These signals either trigger or inhibit the response. Intrinsic inducers are signals that are a response to a stress that caused the cell to become suicidal. This occurs through the binding of nuclear receptors by glucocorticoids, heat, radiation, nutrient deprivation, viral infection, hypoxia, increased intracellular calcium concentrations, damaged membranes, and poly-ADP ribose polymerase. There are two main methods that get carried out through apoptosis. There's targeting mitochondria functionality and directly transducing the signal by adapter proteins to the apoptic mechanisms.
As shown, the adapter proteins were looking for the correct receptors.
As you can see here, we have a cell that has been infected with HIV.
HIV proteins decrease the amount of CD4 glycoprotein marker present on the cell membrane. HIV decreases the production of molecules involved in marking the cell for apoptosis. If there is no receptor, the antigen can't bind to it. This gives the virus time to replicate and continue releasing apoptic antigens and virins into the surrounding tissue.
When a cell receives the stimulus to execute apoptosis, it undergoes organized degradation of cell organelles by activated proteolytic caspases and shows a characteristic morphology. The entire process progresses rather quickly and the products are removed just as quickly. This makes it difficult to see or detect. There are 6 main steps in this process. Step 1. Cell shrinkage and rounding are expressed because of breakdowns of the proteinase's cytoskeleton by caspases. In step 2, the cytoplasm appears dense and the organelles appear to be tightly packed.
Step 3. Chromatin undergoes condensation into compact patches against the nuclear-paranuclear envelope. Step 4. The nuclear-paranuclear envelope becomes discontinuous and the DNA within is fragmented. The nucleus breaks into several discrete chromatin bodies because of the degradation of the DNA. This is known as karyohexis. Step 5. The cell membrane forms irregular bud structures called bleeds.
In the 6th and final stage, the cell breaks apart into many vesicles that have been phagocytized through apoptic bodies.
Dead cells are removed by neighboring phagocytes.
While the cells are dying, they exhibit phagocytic molecules on their surface membranes.